Sawadee Krap, hello and welcome to another edition of the Victor Black podcast. In today's episode, we are going to be talking about the elevation of red blood cell count secondary to the administration of androgens, both in terms of context in TRT therapy and in the use of supraphysiological androgens for performance enhancing outcomes. And today's special guest is Dr. Scott Howell. Scott is someone that I've been following for a number of years now, probably about 3 or 4 years, I think, Scott, since we first touched base.
Yes.
And someone I have a tremendous amount of respect for. Unlike the first 2 guests that I had on my show, Scott is probably less well known to our tribe. I think that's probably fair that your profile is one that is growing in the bodybuilding community. Scott, that's probably fair to say, correct?
Yes, It is.
So can you just tell for people that are unfamiliar with yourself and your background a little bit about both your credentials and your experience within our community, within our tribe itself, please?
Well, my background is in exercise physiology, epidemiology, and mechanical engineering. My last PhD thesis was on androgen toxicity. And my experience in this, what motivated me to study adverse effects was my frustration with hearing things in forums when I was taking anabolic steroids myself. So I took anabolic steroids for nearly a decade. And I went up to some pretty high dosages, and I did have some adverse effects. And so when I come to the realization it was not maintainable at those levels that were suggested, that motivated me to seek another doctoral degree just to figure out the answers. And, that's basically what I did. So I have no ill will towards anabolic steroid users. Actually, I identify with them 100%. I know what it's like, and I've been there, but the important part is that I found is that you'll have a lot of people that are on major platforms and they say there are no adverse effects. Well, that's just denial of the literature. I mean, the androgens are probably one of the most studied drugs in existence. And if you look at all that's been done since before 1935 all the way up to the present day, if I have nearly 40,000 studies in my library, there has to be at least another 15, 20,000 in the world.
Mm-hmm. I agree. It is interesting that so many people would reflect that we don't have all the answers, and I would be the first to agree that we certainly don't have all the answers, but it is an interesting point that you make that there is almost an overwhelming amount of literature for someone like myself, a layperson trying to understand this. It's almost like there's so much information on some of these subjects and sometimes seemingly contradictory information. It's the complete opposite of what a lot of people would support, which is we don't have a lot of evidence to discuss here, but that's completely the opposite, isn't it, in many circumstances?
Well, it is, and the problem with interpreting literature is you have to look for trends in the evidence. So it's not any single study. Any single study can be refuted. Conditions, there can be confounding factors, even in bench work. So what I learned early on is to find trends in literature. If multiple studies are finding things, then I need to key in on it. And so what really obsessed me with looking at toxicity phenotypes was to look at extreme cases where it actually happened. Mm-hmm. And so I was able to map out the specific modes of injury for about 7 toxicity forms. And I don't agree with everything that's been done. There's been a lot of stuff that was like the initial Melchart study in the early 1980s. The fellow applied actual solutions to cells, not the actual hormone. And so what do the solutions have in them? They have benzyl alcohol, they have benzyl benzoate. And so those chemicals alone are directly toxic to myocytes, cardiomyocytes. So when I saw that, I immediately knew there was confounding. But he established cardiotoxicity in that 1983 paper, And so that's when I started going back through all the mammal studies, all the other studies, and literally for like 6 years, I was locked inside of my house. I didn't go out much at all.
Yeah. Cool. All right. So today I wanted to, dive in a little deeper on the subject of, well, really on two subjects, and I tend to consider them together. I don't even know whether that's the right approach. I mean, maybe we could even start there. And that is the subject of the elevation of hematocrit and hemoglobin and, what we would broadly refer to as, red blood cell counts. And we're going to get into some specifics of terminology because I think that's very important because I think it's very misunderstood both in the medical practitioner community as well as our own community, but also the association of escalation of blood pressure. So maybe we could just start with the elevation of red blood cell count, and then at some point we can go back and visit that subject of blood pressure elevation and see where the commonalities lie. Maybe it's the same underlying mechanism. Maybe we could explore that. But if we just start with this understanding that it's my belief that even today that the number one reason that men are withdrawn from testosterone replacement medical practice therapies, as it were, is the concern over the escalation of hematocrit. Is that still the case, that that's probably the number one reason why men are withdrawn from the treatment?
Well, it's interesting because that you bring that up because there's at least 2 or 3 patients that we take on each month that have been at testosterone mills. Mm-hmm. And what they'll do is they'll advise, it's usually a nurse practitioner or some mid-level provider that has a supervising provider that's actually doing the care. And they will actually tell these guys to give a pint of blood a week until they get below a certain level. Well, the problem with that is they always develop iron deficiency anemia and they feel like shit. They have more lethargy than they did when they didn't have any testosterone, really. I mean, they feel like crap. And so it's something that in medicine, we're going to talk about this in a second, but there's a misplacement of polycythemia vera on secondary erythrocytosis, and they're completely different. You have a pathological condition on one hand that not only affects red cells, but also affects other cells. And then you have secondary erythrocytosis from androgen therapy that just increases red cell mass. And so if you actually look at those pathological outcomes like Chuvash or erythrocytosis, I think it affects only a small city in Russia and a small city in Italy. There's a lot going on. There's a lot of metabolic effects that are happening with these diseases, and it's not a one-to-one transfer with red blood cells. So it's even hematologists. If you look at the most prestigious text in the world, Williams Hematology, explicitly in the polycythemia chapter, they say it's incorrect to call androgen-induced changes polycythemia, and that there is no consensus on hematocrit elevations being a risk factor. And I'm going to get to those reasons later on, but after I explain some terminology and stuff like that, we'll rock and roll. But with blood pressure and elevated, with elevated red blood cells, when I think of elevated red blood cells, I always think about elevated hematocrit. So when I say those together, they're interchangeable to me. So the mechanisms.
Can I just interrupt you, Scott? Because I think some of the people listening will be with us, and some of the people we may have already lost a little bit. Can I just get you to, for the purposes of a layperson following this, just explain? So when we undertake testosterone replacement therapy, a significant percentage of men will experience an elevation or escalation of hematocrit levels, of hemoglobin levels, and it's my understanding there is some changes to the iron content within the red blood cells themselves as a consequence. Could you just explain some of this language for us? So what exactly is hematocrit? What is hemoglobin? And can you just start off with a a layman's description of this language and what it means to us?
Oh, yeah, sure. Hematocrit is really a percentage of the solid components of the blood.
Mm-hmm.
And red blood cells make up 99% of blood volume, almost 99%.
Yep.
In a normal person, they have nearly 5 liters of blood.
Yep.
And if you think of the red cell count that they have, if you do the scientific notation, it's 2.5 times 10 to the 13th power cells. And the thing about it is we lose almost 1% of red cells per day. And if you do that in scientific notation, it's 2.5 times 10 to the 10th power. Mm-hmm. So that's a massive amount of cells that are going out of the system, because the life cycle for a red cell is approximately 100 to 120 days.
Yep.
So if we have to redo that every day normally, and then we have androgens that are actually pushing the process up, mm-hmm. You know, it's really a magnificent system to maintain the oxygen capacity for aerobic respiration in the mitochondria.
Without how does hemoglobin fit into that picture? That's something different again, correct?
Well, a hemoglobin, if you look at just a reticulocyte, which is an immature red blood cell, mm-hmm. it doesn't have an iron associated with it.
Yep.
And so there's one iron to every reticulocyte when it matures and enters the bloodstream. Now, you mentioned some other things, but one thing we're going to talk about a little bit later is ferritin. Mm-hmm. And ferritin is a protein that actually takes up iron after it's released from the small intestine into the bloodstream. It takes it up because if it didn't take it up, it would cause oxidative damage throughout the system. Yep. And so each ferritin protein can take up to 4,500 atoms of iron, mm-hmm. and like hold it in a nanocage and control the release of it into the where it needs to go. And usually, if we look at the site of where red blood cells are made, it's in the long bones. Mm-hmm. So it's in the marrow. And there's so many things that can affect that. Mm-hmm. So was that, did that answer the questions?
Yeah, no, I think there are some people that are just going to, I mean, I guess I would classify them more as perhaps people interested in TRT, or they're being prescribed TRT by their doctor that are familiar with these terms, but the language might be a little bit confusing. As I said, we tend to colloquially use terms like, you know, red blood cell escalation or elevation, but as you correctly said, there is an underlying language used in the literature that should be used more correctly. Perhaps I could just show a graphic here. I've pulled out a couple of graphics that I wanted to show as we go through some of these points, and you can speak to them. So this particular graphic is really showing the difference between red blood cells and the other cells that make up our total blood volume. And this kind of somewhat speaks to your point just a little earlier. When we see escalation of red blood cell count consequential to the use of androgens, to the application of testosterone, it doesn't technically fit under the description of polycythemia. That's correct? That's what you're saying?
Oh yes, it does, and for good reason. I mean, polycythemia, there was a paper that was published last year by Borduc, and he actually looked at Chuvash, erythrocytosis, and polycythemia vera. Mm-hmm. And it was interesting because this fellow, he made a call to hematologists to stop using hematocrit as an indicator for therapeutic phlebotomy and actually get to the causal factors. Yep. So if I'll probably jump around a little bit, but
no, that's where we're leading straight into the heart of the conversation. So that's great. Keep going. Yes.
Well, the thing about it is that there can't be a one-to-one correspondence, because if you look at polycythemia vera, and he examined these trials, he examined where the origin of therapeutic phlebotomy or venesection, where it arose, and it actually arose in the year of my birth, in 1978, there was a paper that went out, and it was a retrospective study of 69 patients. And what they established was that, or what they concluded, was that hematocrit decreases might help these patients. Well, fast forward to 2019, and a review of all of the trials that's been done with polycythemia patients in venesection, and polycythemia was actually shown to have higher risk, or wait, venesection was shown to have higher risk than actual chemotherapy. So the polycythemia vera is a myeloproliferative bone cancer.
Yes.
And so chemotherapy is used. Chemotherapy is a huge risk factor for thrombosis. And so having phlebotomy in those patients was a higher risk than chemo itself. So that sort of tells you something. And another thing was that looking at outcomes, most thrombotic events do not occur under conditions of erythrocytosis. It's alteration in other blood cells,
Yes.
not the red cells.
Correct.
So
let me see if I can summarize where we're at so far. So it's fair to say that in the broader sense, many people, as you use the term TRT mill, let's use that term. I like that too. A lot of practitioners running this are concerned about monitoring their patients' hematocrit levels because they see hematocrit as a marker of risk associated with perhaps stroke or some other sort of adverse event that might be secondary to testosterone replacement therapy. But that understanding is somewhat flawed because they are using polycythemia as the risk factor as opposed to the escalation of only the red blood cell count. Is that correct? Is that a fair summary?
well, that's really spot on.
Yeah.
And experimentally, we know that there's a paradoxical response to increased hematocrit, solely due to an increase in red cell mass.
Mm-hmm.
And that's for any secondary reason. It could be androgen induced, it could be hypoxia, it could be fibrosis of the lungs, sleep apnea, etc. And this response does not follow the experimental in vitro conditions that are conducted on static experiments.
Mm-hmm.
And that has been used as a rationale for considering hematocrit as a risk factor. Now, when I talk about a static experiment, I'm talking about viscosity measures made on Oswald viscometers, glass tubes that do not replicate the vasculature.
Yes.
They don't expand and contract. So all these mathematical simulations that are done on that do not correspond to what you see experimentally in real animals. Mm-hmm. It doesn't work that way.
Sorry, I cut you off, sir.
Oh, no. I was just going to say, I found that there's a up to 33% above baseline. So you start at a baseline hematocrit and you increase it 33%. There is an inflection point right at 33% in experimental in vitro living animal studies. There's been some studies in humans, but it wasn't with the hemo concentration I'm talking about. But at that point, there is an inflection, and then blood pressure will rise extremely. And you have to consider that to get 33% above baseline, you're adding, if you started at 45, you're adding almost 15 points to the hematocrit. So you'd be right at 68 or 69. Mm-hmm. And no bodybuilder that I have seen and monitored on anabolic steroids has went past 58. No. Most have never went past 55.
Yeah.
And those that do, they're really anomalies.
Yeah. Okay. So I'm gonna, like you said, jumping around a little bit here. I just want to jump down a couple of short paths to answer some questions that I personally have, and let's start with the first one. So I think it's fair to say, up to a point, it is a plausible discussion to say that the escalation of red blood cell count has potentially a performance-enhancing benefit, not necessarily in our tribe, but in other performance-enhancing drug communities like the cycling community, for example, and they actually deliberately set about elevating red blood cell count through the use of drugs. Have you seen any evidence to say that those practices have resulted in adverse events within those communities as a consequence of their deliberate and intentional escalation of hematocrit levels?
It's odd because with cyclists that categorically, traditionally have used EPO, and EPO is erythropoietin. It's a glycoprotein that's produced in the kidneys that stimulates these different types of cells in the bone marrow.
Okay.
Right, and to kick up the production of red cells. Now, with androgens, we're going to get to the mechanism soon, but it's different. It's
Okay.
similar, different. It's not completely EPO mediated. It's all, the EPO is the indirect mechanism with androgens.
Mm-hmm.
But with cyclists, I have never seen a case report in the literature from anyone that has used EPO. Now, I've heard some stories anecdotally about guys getting dehydrated when they're using EPO. And if you think about it, if you get dehydrated while you're using EPO and your hematocrit is already elevated, okay, people at altitude, sometimes they'll have a hematocrit of 70, 80%. Okay, but they've adapted over a long term. Some develop mountain sickness when they climb higher. But for a cyclist that is not adapted fully to altitude, to take EPO over the long term, if they become dehydrated, that could cause problems.
So that speaks to the idea of there being a separate discussion around relative red blood cell count escalation and absolute. Can you just speak to that a little bit, and the aspect of dehydration and how the consequence of dehydration to our tribe?
Yeah, I had some notes here that I was actually, I want to make sure that I cover when I'm talking about relative and absolute, because I went through this and there were some things that I seen. So when we talk about relative erythrocytosis.
Sorry, can I just put a framework on the table then, and maybe you could speak to that in that context. So the question that I do get asked from time to time is about, so bodybuilders that compete are in a dehydrated state on the way to competition in an aeroplane, for example, are they exposing themselves to things like deep vein thrombosis as a consequence to elevated hematocrit levels and consequential dehydration at the same point in time? These are the types of questions that I don't know how to answer. Maybe we can fold that into there a little bit if we can.
If you look at Flex Wheeler, he had deep vein thrombosis, had to have an amputation. I don't think that was an issue related to hematocrit levels or anything else. I think that was related to vascular disease.
Okay.
So when you have vascular disease, your vasculature becomes stiff. It doesn't vasoconstrict and vasodilate effectively. You lose vascular reactivity, and that's really the onset of cardiovascular disease. When you lose that vascular reactivity, you don't respond to cytokines like nitric oxide and others, then you start to have problems. And from what I gather from his situation, there was definitely vascular disease. Now, is it a risk factor if someone is taking high doses of androgens, they have an elevated hematocrit? To be honest with you, there are several conditions that are conducive to a thrombotic event. Number one, you have to have stasis or reduced blood flow. The second is you have to have a vascular injury. That means it could be a direct insult. You could have an accident or something like that, or it could be a plaque rupture if you had a coronary artery disease. And the third is you have to have activation of a clotting cascade. And so those points in time have to converge at once. And the thing about it is, platelets are the main factor when we're talking about a thrombosis, because a clotting cascade sets off, and then there's a plug that's made. And it's usually to seal an event leading to the outside of the body. to seal blood from going out. But when it happens internally, then there's a big problem. So as far as your question, no, if it's just an increase from red cell count, no, there has to be something else that's there. There has to be an initiation of the clotting cascade, and there has to be some type of injury that would predispose the activation of the clotting cascade.
So I drew you off the topic a little bit there. So going back to the terminology relative and absolute, sorry.
Well, if you think about relative erythrocytosis, there's an increase in hematocrit, red cell count, with plasma changes. So it's the RBC numbers change in respect to the plasma that's there. So if you're dehydrated, that would be a cause of a relative change in erythrocytosis, because the percentage of the liquid portion is not proportional to the cellular components. And it can be due to a loss of intravascular water. And that's the main reason for a relative change in erythrocytosis. Now, if you think of polypharmacy involved with anabolic steroids, diuretics, if you take diuretics and you shift this imbalance, okay, is it going to cause you to have a thrombosis? I don't think so. You have to have the other factors, stasis, where the blood slows. You have to have the insult, and then you have to have the activation of the clotting cascade. And usually when this happens, you have like a hemoconcentration where the red cells are concentrated higher than the plasma. And I want to make sure, does that make sense?
Yeah, absolutely. Really? I guess from again, going back to the layman's point of view, it relates to the fact of, I always tell people when they're going to get their blood work, you need to, I mean, you should always ensure that you're in a well-hydrated state anyway. This is just good practice, but to make sure that, because very often people will prepare themselves for blood work in what we would call a fasted condition for certain of the markers that we need to test, but they forget to drink water. They kind of don't drink anything. And I've seen guys come back with levels and I've gone, holy crap, you need to go back the next day, like drink some water and go back the next day, and it's a completely different reading.
Definitely.
It scares the bejesus out of them. And very, very often, it's just, I say, you know, were you well hydrated going in? They go, no, I didn't have anything from my last meal at night till 12 o'clock the next day when I went in to get my blood drawn. You know, for that whole time, they consume no water. They create a position of dehydration, and they raise their relative levels through that cause. Yeah. So.
Well, you know, that brings us into absolute. Absolute is increase in hematocrit red cell count. That's erythrocytosis. You're actually increasing the numbers of red cells. You're increasing red cell mass. So usually
to the existing plasma volume.
Oh, definitely, and that's why that volume decreases. The plasma decreases because there's obviously more red cells, but usually it's only due to primary reasons or secondary reasons. So primary is polycythemia vera. It's a primary marrow disorder. It's a myeloproliferative cancer. And secondary, it could be due to androgen. It would be acquired secondary, androgen induced. It could be when someone smokes for a decade of their life and they get fibrosis of the lungs, then they don't have the oxygen capacity of the lungs, and then that leads to more EPO production and red cell increases. So you really have to differentiate between these two, because it's really important to call oranges oranges and apples apples, because they can mean different things in different contexts.
Okay. Was there, you said that you had some notes there on relative and absolute. Was there anything else that you wanted to touch on there before I move on?
You know, some of the stuff like I'm looking at now. So if you look at relative, there's a hemoconcentration. And if you look at the studies that's been done experimentally, they either hemodilute or hemoconcentrate to see what the effects are. In the experimental studies I've talked about where they've established that inflection point, they've hemoconcentrated. And what that means is they replicate what would happen during erythropoiesis just to increase the red cell count, not a relative change. So, but if you have hemoconcentration due to decreased fluid, it takes on a different meaning. So you can expand the number like absolute, but when you do it experimentally, it's not the same. And there has to be 3 points. There's a guy, Virchow, that it's called Virchow's triad when we're talking about thrombotic conditions. And some of this stuff doesn't really apply now, but some of it really does because it gives a bird's eye view of really what happens. And again, if a thrombotic condition is going to happen in any real situation, it's going to be when the blood flow is impeded. There's a vascular injury and there's activation of fibrinogen. So when I see guys that are taking tamoxifen or guys that are taking aromatase inhibitors, tamoxifen is an independent risk factor for thrombosis. If you look at the literature, it will cause it in some individuals. Okay, let's just say that someone is dieting down. And aromatase inhibitors are too, but less than tamoxifen. So if someone's dieting down, they're taking aromatase inhibitor or tamoxifen to get rid of excess water or whatever they think is going to happen. And you sort of have a problem because you're precipitating one part of that triad that might happen. So you're increasing your risk when you do that. So my thing is, when I do harm reduction consultations, I look at everything someone's taking. And I look at all the contraindications of that under different conditions. And then I look at their genetics and see, can they actually process that normally? And if they can't, then, I'll give them suggestions on, maybe you might not want to use this. You might want to stay doing this, this, and that. So it's so complicated with bodybuilding because there's so much polypharmacy that goes on.
There's so many factors involved in the discussion. It becomes a very complex discussion. Far more simple, I think, is probably a testosterone replacement therapy thing, because typically we are dealing with, to a lesser extent at least, polypharmacy. So maybe I can ask you if we could move forward to the conversation about the underlying mechanism of action, because I think it would be a fairly obvious point in case to say, look, as we elevate androgens beyond physiological range, we would expect to see consequential to that escalation in red blood cell count. After all, one of the classical indications for use of these drugs is in anemia patients. This is a very well understood historical application. But the thing that I struggle to understand myself is, so why is it that we see the escalation of red blood cell counts at what would, for all intents and purposes, appear to be replacement dosages? You see guys taking 100 milligrams a week, which results in, the very definition of replacement therapy, if you think about it, is, to replace the doses back to where the range of a normal, healthy man. So why does the administration of testosterone exogenously escalate this condition beyond or in excess of natural testosterone production?
Oh, that's a really solid question. For the historical context for this, it was in the 1940s, just after the synthesis of testosterone in 1935, that clinicians actually saw the impact on blood, and they picked up on it. And it wasn't until the late 1940s, after Kokakian had made developed some derivatives, anabolic steroids, that both testosterone and some derivatives were used in the late 1940s to manage anemia. So they knew early on that there was a drastic change in the blood parameters. Now, if you look back And I want to make sure I get the impetus of your question. What was the just the simple gist of the question?
So look, maybe I could show you a graphic, and that will help me explain. There was a let me hang on to open this up. Okay, so this is a graphical representation of escalation of hemoglobin and hematocrit in younger and older men with greater doses of testosterone. I'm sure you know very well at this study. I'm interested in why we see an escalation in hematocrit and hemoglobin levels, particularly in older men, at dosages as little as 50 milligrams a week, which is, very, very much within physiological range. One would even argue it is, quote unquote, not enough sort of androgens. And yet, at even a very moderate dose of 50 milligrams a week, we start to see the escalation of these factors. And I wouldn't expect to see that in older men with that level of serum testosterone through natural production. But as soon as we introduce it externally, it seems to rise. There must be some underlying cause there that I don't understand what it is.
Well, the cause is there's direct hematopoietic stem cell stimulation.
Mm-hmm.
So I talked about the Epo mechanism for a second, but that's only one part. If you look at animal studies, it's pretty consistent that androgens will increase Epo.
Yep.
And they proved this by using an anti-Epo antibody, right, to knock it out. And so it's established in animal studies. But with human studies, there's conflicting evidence. And for example, there's a study by Bachman. I'm going to get to the Coviello study from 2008 in a second. But
Okay.
With Bachman, 2014, he looked at the trial data of the TOMS trial. So if you remember about the TOMS trial, it was actually stopped by an institutional review board because of the adverse events in the treatment arm, mm-hmm. right? And it was older individuals. They received 10 grams of testosterone gel or placebo. And what they looked at was this hematologic response. And the men were over 65 years old. So what they found in the older men of that study that used gel was there was a 7 to 10% increase in hemoglobin and hematocrit. And this was coupled with increased Epo levels and decreased ferritin and hepcidin levels. So one of the things about this study that really struck me was that at month 3, so they're 3 months into the study, the highest average hematocrit was reached. And at 6 months, this hematocrit in older men dropped back down, and it levelized below the levels at the 3-month mark. So
can I just one step back there, because I'm trying to understand this. So it's my understanding that we started to see escalation about 1 month in, and we saw peak levels at about 3 months in, and then we saw like a downward trend starting, after that back down to about 6 months. I guess I'm trying to understand the mechanism. Why would it take 30 days for that to happen? Is that consequential to how long it takes to create new red blood cells? Is that the reason it takes us about a month before we see it?
This all has to do with Epo levels, because if you look at the process of how this occurs, there's a change in hepcidin levels.
Yes.
And hepcidin is a protein that inhibits a ferroportin. And ferroportin is another protein, but it's membrane bound. It's in the intestinal cells, right?
I've just flashed up for for the people watching this, Scott, if I can, a graphic on the screen there and please tell me if you disagree with this. This is, as I understand it, a proposed mechanism by which testosterone increases red cell mass.
Now that was taken from the Bachman study.
Yes, correct. That's
that was
2014, I think. So,
well, I don't agree with all the mechanisms because, hold on one second. So Bachman mentioned 4 indirect mechanisms,
sure,
without really touching on the direct. So for all the listeners, there's basically two mechanisms with which androgens induce erythropoiesis or increase red cell number. That's an indirect mechanism, the androgen Epo mechanism, or the second is direct, which is the androgen induction of stem cells. So what he mentioned in that study was first that testosterone directly inhibits BPM-SMAD signaling in hepatocytes of the liver. That suppresses hepcidin, right? Then testosterone also stimulates adrenal secretion of EPO, but he states that there are unknown mechanisms that stimulate erythropoiesis, which could suppress hepcidin. But that's incorrect, because if you actually look at the literature, this group that put out this study is from the Boston group. They're affiliated with Harvard. Basine, he was the one that conducted the trials that Coviello looked at. And Basine actually conducted the study, the Tom's trial that we're looking at the secondary data analysis about. So what I've seen is that there's a large amount of literature that is ignored by this group. So the third mechanism that he actually put on here was that testosterone will upregulate the expression of GATA2 or GATA1 and GATA-dependent genes, which could increase the Epo sensitivity. Well, he's getting close to the mark, but it's not these genes that increase the sensitivity. The androgens themselves indue these cells to be Epo responsive. And the last point he put was that testosterone converted to estradiol can regulate hepcidin transcription. Well, the problem with this is that we know what occurs with EPO, and we also know how androgens directly stimulate these stem cells. So this is only partially what I would attribute. Now, some of the mechanisms like the liver, yeah, that could be the reason for it to do it, but there are some things that we should really discuss because there's discrepancies.
Correct.
And if we find the commonalities, then we're confident.
So can I apologize. I'm trying to understand, but I still don't understand the point. So maybe I can ask the question again, because quite bluntly, I don't know who else to ask these questions to. So maybe I can pose the question. So let's create a situation. Let's say we have a hypergonadal man who's in his mid-40s, and he has currently serum testosterone levels in the, let's just say, the 200 nanograms per deciliter range, and he's experiencing during a number of less than ideal life experiences, and he's a prospect for testosterone replacement therapy. Okay, and he is provided with a dose of testosterone that raises his serum testosterone levels. So let's just arbitrarily say 600 to 700 nanograms per deciliter. So well within physiological range. Yeah. A month later, we would start to see escalation of red blood cell count, which would peak in about 3 months. I still don't follow why we would see in that individual escalation of red blood cell count, as opposed to another healthy man who was walking around with serum levels at of the same level. Why does the administration of external testosterone cause that event? I completely understand once you step outside of physiological range, it's how low it's a dose response. The more you put in, the greater you get. But it seems to be, and maybe I'm completely missing the point, it seems to be simply the act of administering androgens through some external mechanism has a impact on these markers.
Well, it does.
Okay, I'm missing the why. That's what I'm trying to understand. So why?
Okay, so here the why is that there's direct stimulation of these progenitor cells. So there's a BFU in the bone marrow, and that BFU is burst forming unit. It's stem cells. They're not EPO responsive. They don't have EPO receptors. And then when they transition the next step to a CFU, it's colony forming unit. And so the difference between those two units is one is responsive to EPO. They have receptors. The other is not responsive. So androgens directly impart the ability of those blast forming units to have EPO receptors. And so they transition to a colony forming unit. They're responsive now. So what actually happens, the direct mechanism is that when testosterone is reduced in the bone marrow, and even anabolics, even derivatives, they're reduced to 17 keto steroids. And that, upon binding, it increases the production of mRNA for non-differentiated marrow cells, and that is Epo sensitivity. So the actual Epo gene gets turned on, they start expressing these receptors, and then they're responsive to it. Once they're in the colony forming unit, then any circulating level of Epo can stimulate them to mature. They'll turn into reticulocytes that don't have the iron yet. They're still immature, but then they will actually associate, if there's enough ferritin available, a heme atom, an iron atom. So that is direct hematopoietic stem cell stimulation. So the indirect mechanism just goes on Epo to do it all. And in normal conditions, it would. When you introduce androgens and they get reduced to 17 keto steroids, that is the stimulus that turns up the Epo gene. And the interesting part, though, is that once these red cells become reticulocytes or mature red cells and enter the bloodstream, they don't express Epo anymore. They don't have the Epo receptors. So they don't have them in the beginning, then they're given the ability, they have them for a short amount of time to respond to Epo, and then they lose that ability through the maturation process. Does that make sense?
Yeah, now I understand the missing piece of the puzzle. So that leads me to the question then. So the various testosterone derivatives that have been evaluated over the years for the treatment of anemic conditions, because it was, as I said, historically a fairly common clinical practice to use these derivatives in that capacity. Why do the different derivatives have a greater or lesser stimulatory effect on production in this regard? So, for example, something like equipoise or boldenone is remarkably similar in chemical structure to the parent molecule testosterone. It's not vastly different, but it does seem to have, at least anecdotally, a significantly increased effect in this regard. Like, you know, if you put them in a dose response, comparison study, I would predict that we'd see a greater escalation of hematocrit and hemoglobin from boldenone than we'd for a milligram per milligram dose of testosterone. Given the fact they're so remarkably similar in sort of molecular structure, why do we see that difference?
Well, it goes back to actual configuration of the molecule. So there was some in vitro work by Nachels in 1969 and Gorshin in 1970 and 1974. And what they established was that androgens with a 5-alpha hydrogen position over the top, these stimulate renal and extrarenal Epo production, whereas androgens with the 5-beta hydrogen under the bottom, they enhance the differentiation of stem cells. So you have a split here on how the molecule is in a three-dimensional sense, not flat. So when we see the steroid nucleus, A, B, C, D, and the carbon rings, all that stuff, we have to look at it three-dimensional to understand it. And so I dug so far in the literature to answer this, it just really made my brain hurt to get to it. But what I found was that even with knowing this, if you look at like the schematics that are on like ChemMed, PubMed, and stuff like that, you can't really tell if there's a 5-alpha hydrogen configuration or 5-beta hydrogen. It was something that I had to dig into further, but there was experiments and they went through different cell cultures to see how they would respond. And what I've seen with guys that I've monitored is that some compounds will jack the hematocrit up. Yep. Some will not. And in the US, only decadurabolin and oxymetholone are used for anemia. Well, so what makes them so great? Well, even with this definition on why there's a differential response, it's not clear, because I really didn't have enough time to look up to see if decadurabolin was in the 5-alpha or whatever. But this explains a lot of variation that I've seen personally with guys that I've monitored blood work on, that I looked at all this stuff. But it sort of makes sense when we think about it in the big picture perspective. We know there's a reason why there's differential effects. I mean, it's just like the whole reason that androgens were created in the beginning, synthetic androgens by Kokagian, was that he established nitrogen retaining ability. And he did this in 1936. It was a year after testosterone was synthesized. And he used androstenedione. It used to be a supplement, oral, that was available in the United States until the ban. And what he's seen was when he injected this into dogs, they had a higher nitrogen retaining ability. And so that led him for like 60 years to study the ability of these androgens to actually stimulate mineral retention. So if you look at adverse effects, if you look at the beneficial effects, they're mediated. All androgens are characteristically have nitrogen retaining ability, but it varies. We know it varies with different compounds. So we're remiss if we say that other effects don't vary, because they do. They all have them. It could be smaller than others, but we know that they're there.
It's completely off the subject. I apologize. So I don't want to drag you off the subject for today. But would you say that, because my own personal opinion is that, you know, the degree of myotrophic effect of these compounds is quite profound in the bioassays we have in Hirschberger assays and rat models and things like that. But I don't see this in human studies. In human practice. So the adage that so many milligrams of, you know, Anavar, for example, is 3 times more potent as an anabolic agent than testosterone. I don't see this in practice, and I've never seen anything in the evidence to support this idea. But I do agree with you that there is very good evidence that there's very large differences in nitrogen retention between the compounds in human studies. So maybe one day we could get onto that subject and explore that further, because I don't want to drag us too far off the subject. So what we've basically gotten to is that we understand the underlying mechanism, why it's happening. We understand that it's something we see in a dose responsive capacity. So the more androgens we introduce, up to a certain threshold at least, we see an increase in these markers. But as you pointed out, and even that study that I showed you with a greater dose of testosterone, although we see a relative increase in terms of dose response, there does seem to be a point whereby even at 600 milligrams of testosterone, we're still within what is considered generally to be therapeutically acceptable, that range, which doesn't seem to be a high risk. So I guess the next question for myself is, when we have recreational users of these compounds, you know, do you have a cutoff level? You say, look, you know, between this range and this range, we can live with that, but there is a point where we need to, you know, take a line of action. Do you have a marker? Typically, that's basically represented as kind of like, you know, 0.52% sort of.
Yes. Well, the threshold is the inflection point. Mm-hmm. So the inflection point that's consistent across all the experimental studies in vivo with live animals, mm-hmm. It's 33%. Yep. So if you take the percentage of your start line, starting hematocrit before going on testosterone or anabolics, whatever, yep. And you calculate 33% of that, added on top of it, that is where you do not want to hit, okay. Because that is where this whole thing comes in. And it's, yep. The whole thing about how clinical practice has missed it. Mm-hmm. They've modeled on static experiments, but in real life, in real animals, it doesn't do that. It will increase because the vascular beds, the cardiac output increases and makes the body more efficient with a moderately increased hematocrit. Mm-hmm. But when you go beyond that and you get to almost that threshold, you see an inflection point where viscosity is exponential. It rises. And with viscosity, obviously, blood pressure is going to go through the roof. Mm-hmm. So the only time that it's really detrimental is when the hematocrit goes through the roof. And that would be if someone started at like 45 hematocrit, they would add almost 15 to the top of that. They would be nearly 69.
Mm-hmm.
That is where the inflection point would increase.
Okay,
But
so
I've never seen that.
Okay, I just want to go back to that graphic. This is a graphic from that same study, the effects of greater doses on testosterone, and this shows a percentage increase by greater doses of hematocrit and hemoglobin. So you can see here in older men, even at 600 milligrams a week, we can see a percentage change in let's just say of the magnitude of somewhere between 10 to 15% increase. So what you're suggesting is greater than 30% is of concern to us. Is that correct?
No, I'm saying that that's where the inflection point lies.
Okay.
And you have to actually calculate or estimate or assume that animal models don't respond to human models. Mm-hmm. So a safety margin has to be applied. If you look at the Coviello study with those graded doses, there was a typical dose response. It was in both groups, but what happened was the older men had higher total hematocrit, but they slowed. The younger guys increased hematocrit really quickly up to a month. And then the older guys, they didn't get up to that level, but they started increasing, and they kept on increasing. They kept on increasing to 60 and 90 days. Mm-hmm. And then they had top outs that were right below 50. And the young guys were right at 45 average across the board. Mm-hmm. So the age-related difference, I can't speak to. We see it. We know it's there. It's paradoxical to the Bachman study because the Bachman study showed completely different. And we have to look at the mode of administration. The population was highly comorbid. So in these individuals, for these two studies, the younger guys were otherwise healthy, and the older guys were otherwise healthy. Mm-hmm. Otherwise, or regardless, they wouldn't be given 600 milligrams of testosterone per week if they had that. So maybe the mode of administration here is one thing that shows the differential response, but the Coviello study.
Can I just ask you to explain that? So, because I understood that it was by intramuscular injection for both groups, correct? There's the same pathway of administration. Why would I miss that point there? Why would that change?
Well, if you think about gels like Androgen, they get into the system and get out quick. There's not ester that's controlling the release. So anytime that you have esterized testosterone, it is released, and the ester, when it's cleaved, it has to go through hydrolysis in the liver, and things happen. So there's downstream effects. And what I see with the Coviello study is a more accurate depiction of what we see.
I understand your point now. So I meant between these two cohorts in this study, it's the same administration pathway. You're talking about the two different studies. I understand. Yes. Sorry, my apologies.
So older and younger, we do know that androgen receptors will express higher in older individuals. Mm-hmm. It's something that I've seen in several experimental studies, and to be honest with you, I do not know a mechanism for why that younger individuals would respond higher. It could be that there's higher sex hormone binding globulin. Mm-hmm. And there's not there's a differential in the free testosterone, what's bioavailable. So it could be that younger guys have lower SHBG. Mm-hmm. It could be that the older guys have higher. It's several factors. And I remember reading the discussion of the authors in Coviello study, and I believe that they had mentioned sex hormone binding globulin and some other factors, but they really were just cursory. There's not, I don't know a reason for that. We know it happens. Why? I'm not sure. And it could be that hepcidin in older individuals takes a higher dose to be suppressed. Mm-hmm. And if you look at the Bachman study, hepcidin was suppressed at 1 month. And if you think about the suppression of hepcidin, when hepcidin is suppressed, then that means ferroportin can offload all this iron to ferritin to incorporate. So when it's non-suppressed, it actually inhibits ferroportin. So these fluctuations that we see are expected because it's a dynamic system, and the human physiology needs to keep that 2 times 10 to the 10th power cells per day in production. So it could be amplified. How was that?
Yeah, no, that's fabulous. I think unfortunately, it's my nature. I think I drew you off the track. So I just want to back up one little notch. So we were talking about, so a generalized framework or recommendation to individuals that are seeing escalation of hematocrit from baseline position, in other words, without androgens, as a otherwise healthy natural male, and we introduce androgens, it's a reasonable expectation that we will see an escalation or an elevation of these markers. Obviously, some degree of elevation is, I think, accepted, but there's a threshold that we mentioned. Can you just speak to that? Because you were kind of getting there. I think I cut you off. I apologize. You were saying that there's an inflection point at about 30% of change. Can you
Yes.
say, so if that inflection point is about 30% and we need to have a safety margin, as you mentioned, where would you place that? What would you say is a guideline in terms of us monitoring our markers in that regard?
So if someone has a starting hematocrit of 45, then 33% above baseline is around 60.
Mm-hmm.
And that's what, that would be the critical point.
Yep.
Before the hyperviscosity and the blood pressure increases, et cetera.
Mm-hmm.
So, I've always err on the side of safety, and it could be, the first thought in my mind is to decrease that by 25%, 25% of 33%, 1/4. But if someone that wants to be really studious about it, take 1/3 off the top of it and make it 22%.
Yep.
And so if you do that, then you will see what we see in practice. See, I've never seen many people that are above 55 to 58, but it's usually around 55. And if you knock that down another, take 11 off the 33, then you get hematocrit that's around 55 to a little bit above.
Mm-hmm.
So it's all contingent on the starting hematocrit, though.
Yeah.
So if the individual starts with a higher hematocrit at 50, then that number is going to be higher, and their safety margin has to be a whole lot huger. It has to be bigger because they're already starting at a high amount.
Agreed. And there's just for people watching this, I just wanted to point out there is let's see if I can find the graphic I want to show you. This is a graphic from a study that speaks to the genetic component that's at play here. So there is like pretty much everything. You like it?
That's Sitzman. I love
agree?
you. Yeah.
Do you agree with the study that shows that there is a genetic component here?
There is, and it's predicated on a polymorphism. And this polymorphism is responsible for 40% variation in response to androgen therapy from individual to individual. And there's a substantial ethnic or racial variation in response as well. So if you look at all the studies that's been mapped out, and Sitzman and Nieschlag in this study, I think it was either 2001 or 2006, I can't remember, but they mapped this out, and it really shows what we deal with in clinical practice on a daily basis. You have to treat, in this article, in this study, Sitzman concluded that there is no normal reference interval for any single male. And that's true because there's a 40% variation. So a normative reference interval based on a population of individual is not, it doesn't apply. So this 40% variation in response is huge. And individuals with a longer length of this polymorphism, they have to be titrated up and up and up, but then their side effects go up and up and up. So it could be that it also stimulates erythrocytosis. And I think there was, he charted that out in that study.
Mm-hmm. Mm-hmm.
Good
Okay,
stuff.
so I apologize. I really want to touch on a couple of other points before we run out of time. So I apologize for jumping a little bit. But
No, you're good. You're good. It's been
so
great.
the I guess the next two areas I wanted to talk about is the practice of phlebotomy, the practice of removing blood volume, consequential to individuals seeing, what I would consider to be marginal sort of elevation of hematocrit levels. I tend to steer guys away from that. I'm not a fan of phlebotomy as a treatment methodology for our tribe. Can you speak to that? Do you agree with that? Do you disagree with that? Would you see phlebotomy as just something that we really just don't need to be doing?
Well, phlebotomy across the board has been misunderstood historically.
Mm-hmm.
Phlebotomy actually came into the medical literature in 1978, the year of my birth.
Mm-hmm.
And basically, it was a study by Pearson, and it was a retrospective chart review of polycythemia vera patients.
Mm-hmm.
And what they concluded at the end was it might be beneficial to reduce the hematocrit in these polycythemia vera. Now, once again, let's remember polycythemia vera is different from secondary erythrocytosis. There's more than just a red cell increase. There's increases in other cellular components. It's a cancer. There's changes. The morphology of the cells aren't the same. So there is no one-to-one correspondence. So what they concluded here was that venesection or therapeutic phlebotomy might be beneficial. It took off like a shitstorm.
Mm-hmm.
And then now every hematologist that you talk to, they have a dogma that's already in their head,
Mm-hmm.
and it's because this has been perpetuated.
Mm-hmm.
So the quick answer is no, it shouldn't be, unless you have risk factors for thrombosis that are real risk factors. Tamoxifen, if you have a familial disorder or something like that, no. We receive a lot of patients from these T-males that do this therapeutic venesection, and they end up causing iron deficiency anemia.
Mm-hmm.
And that is just something that people don't want to experience because you really feel like crap. You don't have any energy. I mean, it's not good. But we can rectify that pretty quick because of the life cycle of the red blood cells. We can kick it back up and get it going again. But it is not anything that is, it's not valid. The whole practice of therapeutic venesection by practitioners with testosterone therapy is completely inaccurate. It's misguided. It's not aligned to the literature. If you look at the studies that have been conducted on polycythemia vera, there's been trials that's been conducted that compared chemotherapy to therapeutic phlebotomy. And what they saw was, well, chemotherapy is an independent risk factor. It is. So they wanted to compare them. Therapeutic phlebotomy was a higher risk than chemotherapy. And that's just outlandish to see that, because the whole idea to give blood to decrease hematocrit is predicated on polycythemia vera. So what you have now is clinicians that can't differentiate between the two, although they know there's a JAK2 test that they can do to rule it out. But it's one of the things where if you want to lose a beneficial effect of the androgens you're taking, don't use it. Phlebotomize. Get those red cells out and don't have that extra iron or oxygen capacity. Just drop it. So it's like a reverse consent form, really.
So I know that there is a study that I'm aware of that actually looks at the practice of phlebotomy and the administration of androgens and the consequential re-escalation of I think in the particular study I'm thinking of, it's they're measuring hemoglobin levels. One of the great questions I've always asked is, so if I understand the idea of removing blood if you remove the stimulus, but if you leave the stimulus in place, then removing excess blood volume, it's just a matter of time before it comes back. Have you ever seen anything in the evidence or even just through observation that talks about if you took, let's say, a pint of blood out, how long would it take for that to go back to the previous level? I'm assuming that, like I see these guys that almost by practice, remove blood every 12 weeks. Like they literally go in and every 12 weeks have blood taken out. And my argument is, I say, well, I don't think that you would, last more than a week before basically your body is replenishing those blood cells because you haven't removed the stimulus. Let's set aside everything you've just said about like best practice and saying so even if it was an acceptable practice, and I completely agree with what you're saying, if you take out a 600 ml of blood and you leave the stimulus in place, it's just a matter of time before it effectively replenishes itself. Have you ever seen how long that takes? Because there is a study that shows that is true, but that particular study doesn't speak to duration of time. Do you have any insight into that? If we were to do that, how long it takes to come back?
Well, I'm not really
sure. Show the study I'm talking about. You might know. I'm sorry. I think people do get some value out of this. So this is the study that I'm talking about, where blood donation and testosterone replacement therapy, and it looked at donor characteristics and hemoglobin measurements, but it doesn't have like multiple. It doesn't look at it in time ranges. It just simply says, look, when they come back, they have the same problem over and over and over again, basically.
Well, the issue with that, Victor, is that under androgen therapy, you reach a threshold.
Mm-hmm.
And there's something that happens when you reach a threshold of hematocrit where it don't go past it. And what I mean by that is I followed guys for years that was taking gram doses daily, or not daily, weekly.
Yep.
And their hematocrit reached a point where it didn't go past that.
Agreed.
Now, if you think about it, there's a lot of mechanisms in physiology. If it's overused, like a pathway is overused, it will start to shut down. Mm-hmm. It will start to like adapt to whatever's in front of it. Like CYP enzymes in the liver, they sometimes when they get really taxed, then things will start to like change and alter. And so with this, what I've seen, and this is, I've probably looked at least 300 or 400 guys' lab work that have used anabolic steroids. And what I see is there's a point where it doesn't go past that, no matter if they do therapeutic phlebotomy or don't. Yep. And I know that myself, I didn't really move past a hematocrit of 52 on about 5 years continuous use. Mm-hmm. It didn't go past that. Yep. It went up to that point, and there was times that I did a therapeutic phlebotomy when I didn't understand the literature. But I guess what I'm trying to say is that if you look at the life cycle of a red cell, and it has to go out, and you think of the life cycle and the maturation process, the information to answer this question is how long does it take for androgens to stimulate that erythropoiesis and make a mature red cell?
Correct.
And I've never seen that in the literature.
Okay, great.
So.
Okay, cool. So one last key subject, if I can, and that's the subject of one of my favorite subjects, which is the use of ARBs in individuals that are using supraphysiological levels of androgens. And I wanted to basically just show you a graphic here and get some feedback from you, which basically talks about the fact that ARBs, in this particular case, we're looking at two ARBs. One is losartan, and the other is irbesartan. And the fact that both of these compounds are basically hematocrit modulators in their own right.
They are.
So
and a lot of people don't know that.
Yeah, it's not well done. And this is one of the reasons that I say to guys, look, there's like a list of reasons that I think that everybody that's using super physiological levels of androgens should simply use an ARB by default. And this is one of them that you can forget about this. You can see on this graphic here where there are, just for anyone reading this, these individual black lines represent individual people that start with what we would describe as elevated hematocrit levels, and through the administration of losartan over a 3-week time frame, you can see the gradual decrease in hematocrit levels over that time block. Would you agree with the premise that ARBs are a useful tool for many reasons, including the modulation of hematocrit in this sense?
Well, I agree that ARBs, and really if someone doesn't have access to ARB and ACE inhibitor, are really beneficial to bodybuilders. And the reason why, we've known for 30 years now, 30 years of literature has shown that androgens in the doses that bodybuilders take stimulates angiotensin. It activates that system. And so there's so many pathologies that are related to angiotensin too. I mean, half of the toxicity forms that I found that I was able to name in the literature, they're mediated by angiotensin 2.
Mm-hmm.
And so it is only good prophylaxis, preventative measures, to take an ARB if you're gonna use high dose androgens or any like real moderate doses of androgens above therapeutic testosterone. If you look at the cardiac pathology, if you look at the renal pathology, if you look at hypertension, there's several things that mediate hypertension, but if you use an ARB, you're really attacking the source. You want to look at the cause of it. And so the only thing outside of angiotensin that I've found that develops a pathology is 20-HEAT. It's an icosanoid that's a metabolite of arachidonic acid.
Mm-hmm.
You know, you've heard of arachidonic acid before. Well, this metabolite causes major destruction in some parts of the body relative to high androgen doses.
Mm-hmm.
And so it works paradoxical. In some organs, it will like make the blood flow great. And then the vasculature in some other organs, like the kidneys, it'll make it go to shit.
Mm-hmm.
And so the thing about it is the culprit behind 20-HEAT is angiotensin 2. Mm-hmm. So you hit the cause. You don't wait for this pathology to occur, because in all the animal studies that's been done where they used the angiotensin blockade, they blocked the angiotensin out, cardiac pathology did not occur. And this was with extreme doses of androgens. It was bizarre when I had seen all the androgen blockade studies and no one had ever talked about it. It was just beyond my conception at that time that no one had ever voiced the concern in the bodybuilding industry that angiotensin, that's the #1 killer of bodybuilders.
And it's quite ironic because most people in our tribe try not to use these drugs. They see them as like a last line of action, and it's quite ironic that I see it as like, that's a default position. If you're going to go down this pathway, that's on the table from day one. And yet the culture is you should do everything you can to avoid using these drugs and only use them as a last line of action, which is, this is why I'm so passionate about so much of the information that you read online I don't agree with, and I try to correct, because it's just like, well, hang on a second, that's it's not my opinion, it's completely the opposite of what you're saying. It's not even like, well, there's shades of gray, we could have a difference of opinion. It's no, you're saying don't use one, and the reality is you should be pulling them out by default from day one as one of the primary preventative measures against long-term adverse events of this. And this is why it's so frustrating for me in this community, that is, you read these things and you just go like, that's just completely the opposite of what we should be. Right. Phlebotomy, don't do that. ARBs, you should use that as default. It's very, very frustrating for someone like myself, and it's so empowering to find individuals like yourself with the credentials of the education that can help me understand this better and other people who understand these things better. I do, however, want to go back to your point. You mentioned about arachidonic acid. I have never heard that before. I know that there are a number of arachidonic acid supplements that are being promoted to the bodybuilding community. I won't mention names, but are you suggesting that by extension that those supplements are things that we should probably avoid because the metabolites have the potential to cause harm? Am I misreading that?
No, I don't say that. The 20-HEAT protein, or 20-HEAT icosanoid, I'm not sure if there's any correlation between if you ingest a supplement of arachidonic acid, if it actually goes, because it's liver produced. There's CYP mechanisms, the P450s in the liver they have to convert this.
Okay.
And I'm not sure, there's been no studies I know of that have used arachidonic acid and did the metabolites. It could have paradoxical effects. So I can't say with 100% certainty that there's anything there. And I only know that from the literature, it's an arachidonic acid metabolite.
Yeah.
But the effects it has downstream, if it's in the wrong situation, under normal conditions, 20 heat modulates the vasculature, the pressor responses. It does vasoconstriction, vasodilation differently in different organ systems, and then the vasculature and like the periphery. When high dose DHT, and really high dose DHT is applied, then that turns on its head. It reverses, and then it's like a potent vasoconstrictor of the vasculature. And you see some individuals that are on anabolics that can never get their hypertension in check. And the only thing I've seen probably 50 individuals out of thousands of individuals that I could not ever nail down their reason for hypertension. But I do, after seeing the data on this is a viable reason. It's androgen-induced hypertension from 20 heat. It is a viable mechanism. It is there. It's been well documented in experimental studies and then in vivo studies. And the thing about it is what it confuses people is that it has paradoxical effects in one tissue compared to the other. So in one organ, it might be renoprotective, cardioprotective at normal doses. But then when you escalate the dose high, then it's not the same. Then it turns like an evil cousin and starts destroying tissue. So,
wow, what can I say? That was a very interesting hour and a half. I learned a great deal. I thank you so much. Listen, rather than drag this on too much, maybe what we could do is I can ask you to explain a little bit to the listeners about the Tier 1 Center for Research. Specifically, what I'm actually interested in is I have a number of people who are medical practitioners that I know follow my content. They are people who run testosterone replacement clinics and things like that. You provide an educational or consultancy services to medical professionals that are looking to learn more about this so that they can better advise their own patients. Could you just explain a little bit more about what Tier 1 does and its role in our community?
Well, Tier 1 Center for Research is part of Tier 1 Health and Wellness. We separated these two parts of Tier 1 because of my experience and my expertise has always been in illicit androgen abuse. So I had to go in that direction. It's sort of hard to mesh something with clinical practice if there's a discrepancy. So with Tier 1 Center for Research, it's about changing a health paradigm. Guys will use anabolic steroids. I know that, but can you use them safely? Well, the only way to use these drugs safely is to have your genetics sequenced. We need to look at target genes. We need to look at different aspects of androgen metabolism. And these things are correlated with adverse outcomes. And so the most educated bodybuilder in the world today would be one that had their whole exome sequenced. And they would have these target genes that we know influence androgen metabolism, and they would have them sequenced to see where they fit in that continuum. Because we know anecdotally, we see some individuals that can take a low dose of any androgen and gain 10, 15 pounds of lean mass. They just look completely different. They respond. Well, why? There's a reason why they respond, and it is based on some polymorphisms, and we can map that out now. And so with the Tier 1 Center for Research, I have a soft spot for guys that have been locked into the bodybuilding lifestyle, and then they start to see health consequences. And that's really what drives me, is to get these guys up and running. Try to prevent any more harm, try to do the best with what is there, and then rock and roll. But the Tier 1 Center for Research is, we're studying EDCs here, endocrine disruptors, and things like that. But the thing about it is, there's an educated way to use anabolics. And if you're not aligned with the current literature, you could be making mistakes that will affect you the rest of your life. And sadly, I've seen that in so many guys that make the wrong choices early and then keep on going. And so that's what we're here for. And we also have the Tier 1 Center, the Tier 1 Health and Wellness, where we do testosterone replacement therapy and hormone replacement therapy for men and women. But this is a completely different population than what I deal with. It's people that have a genuine deficiency and want to get their health back on track. So thank you for doing that. Thank you.
That's yeah, that's awesome. As I said, a fabulous hour and a half. I'm sure a lot of people had their beliefs and conceptions challenged a little bit there, and it was fascinating. Obviously, I mean, I've mentioned this to you several times. I'm hoping that we can get you to come onto the show on a regular basis, because there's so many things I want to talk to you about. It's just a case of scheduling, because I know you're incredibly busy with the many things you do. So maybe maybe I'll leave you alone for a month or so, then I'll come knocking on your door again and ask you to come and share your expertise.
I can say this to anyone. I've since I have interacted with you for the past 3 or 4 years on Facebook, you're one of the only people that listened to some of the concepts I was throwing out. I would send you studies and things like that, and you listened and embraced it and looked at the data. So we're in a world right now that looks at that cannot vet evidence. And so the thing about it is, and what I like about what you're doing, and it's interesting because I think you're probably one of the best performance enhancing guides in the world, because you look at the literature, you don't make decisions based on BS or anecdotes. And so you're guiding people by evidence. That's how it should be. And so, you're making a mark on people, and that's what it should be. One mind at a time is what I say. Sometimes it's really slow, but if you can motivate anyone, then you're good to go. And I appreciate you having me on. Thank you.
Thank you. Thank you very much. All right, we'll wind that up there. Thank you, everybody, for listening. Thank you, Scott, for coming on. And I will look forward to seeing you at the next Victor Black podcast. Saudi Cup.